Well, good afternoon for this last session. Last but not least, because transmission remains a big problem. And the first presentation will be given by Dr. Sofie Braet from the Institute of Tropical Medicine in Antwerp. You have the floor, Sofie.
So I will present an update on the STARLEP study. It's a study funded by Leprosy Research Initiative and the Turing Foundation. And the STARLEP is an acronym for Study on Transmission and Resistance of Mycobacterium leprae and lepromatosis in Africa. I will first give you a background of leprosy in Africa. So in DRC, Burundi, and Ghana and Cameroon are the countries where this study will be conducted. They all have reached the elimination target, but case detection remains stable through passive case methods without microbiological confirmation. Ongoing transmission is suggested by the high number of multibacillary cases, 81%, childhood infections 10.6%, and grade 2 disability of 15.3%. This indicates, like I said, ongoing transmission and also a diagnostic delay. Drug resistance surveillance is lacking in the study countries, although WHO recommends to test in 10% of the multibacillary cases. But up to date, only 6 African countries have conducted some testing, but also not country-wide testing in Africa. Also, the role of M. lepromatosis, the second causative agent of leprosy, has already been detected in Asia and in the Americas, but the contribution in Africa is not known because there is no screening for it. So limited countries have tested for it. And then genomic research is limited due to the culturing issues in vitro. So passage through mice is necessary to do whole genome sequencing, or you have to do targeted capture to capture out the leprae, and there are only a few labs in the world to do that. And the WGS that is available for leprae worldwide is under 1,000 strains, and the African contribution of that is very low. So we want to a bit cover that gap. So the research question is drug resistance and lepromatosis infections are contributing to ongoing leprosy endemicity in 4 African endemic countries. So the primary objective is to estimate the proportion of leprosy due to leprae and lepromatosis, and also due to drug resistance in each study area. And secondary objectives are determining the proportion of molecular confirmation among clinically diagnosed leprosy patients. And then on the ones that are molecularly confirmed, we will try to do whole genome sequencing for those possible. For those not possible, we'll try to do duplex mixed-lab. And by doing that, we will try to identify genotypically clustered strains within a community and try to identify genes under positive selection, which might be linked to drug resistance. And another secondary objective, to my opinion, a really important one, is building a network of externally assured partners in leprosy endemic countries with the molecular capacity to support leprosy diagnostics, including knowledge transfer, capacity sharing, sustainable dissemination of results. So the methods of this study are patients will be identified in skim camps or passive case finding, or more active case finding by contact screening, depending on in which area we're working. Then they will be referred to be diagnosed based on WHO guidelines, and then eligible patients will be asked for informed consent, and a 4mm skin biopsy will be taken. So study implementation will be done by the labs in collaboration with the national leprosy programs. So the national leprosy programs will be trained for the 4 mm skin biopsy sampling, and then the samples will be transferred to designated laboratories in each country. And Burundi will initially rely on the ITM in Antwerp until the platform to confirm leprae is implemented in the country. So the sample size and feasibility. So the WHO asks for 10% of the multibacillary cases to be tested for drug resistance, and this is compiled 400 patients a year. And this study will serve as a pilot to demonstrate feasibility and advocate for broader screening efforts across the participating countries. So in Burundi, we will work in 6 health facilities in the Rutana province, and we will aim for 80 to 100 patients a year. And testing will be done in Antwerp until biomeme is implemented in Burundi. In Cameroon, they will aim for 50 out of 150 patients diagnosed per year across entire Cameroon to be tested in Pasteur Institute in Yaoundé, and sequencing will be done in Antwerp, but training of sequencing to be transferred to Cameroon. For that, extra fundings will be sought. And in Democratic Republic of Congo, Kinshasa and Congo Central will be included, and there we aim for 80 to 100 patients per year. Testing will be done in INRB, and sequencing will be done in Antwerp. In Ghana, it's a countrywide study, and they aim for 200 patients a year, and they will be tested in the Noguchi Memorial Institute of Medical Research in Accra, and sequencing will be done until sequencing will be implemented in Ghana as soon as possible. But for that training, we need to find funding as well. So we will prepare patient sampling bags where all the consumables for sampling a skin biopsy, a skin slit smear, if that's included in the routine care, and a nasal swab will be included as well. And then in the lab, the biopsy will vertically be cut in two, will be extracted with the biomeme DNA extraction technique, and then that extract will be used to do biomeme qPCR, which is a portable qPCR system that works on batteries. And part of the DNA extract will also be used for sequencing. So this is the whole genome sequencing workflow. So in the beginning, you will have a whole DNA extract, which contains more than 99% human DNA and less than 1% pathogen DNA. The DNA will be fragmented, it will be end repaired, adapters will be added, and then you will amplify the DNA. And then on this amplified so-called library, we will add biotinylated probes that will hybridize to the pathogen DNA. And these are probes designed by Roche Diagnostics. And then by magnetic beads with streptavidin, the leprosy, the lepra DNA that is being bound to the probes will be fished out, and then we will only work on the fished out lepra DNA to do the sequencing on. So a short update on where we are now. We did a lot of preparatory work with grant agreements, data sharing agreements, material transfer agreements, insurances, protocol development, data management plan, informed consent development in different languages, SOPs development, database development for capturing all the questionnaires, and the protocol and ICFs have been submitted to all the countries already, and recently, last week, Cameroon and Ghana were approved, and DRC was approved in December last year, and Burundi is submitted, but not approved yet. So we're still waiting for the approval for the start of the study, and therefore we also didn't order the consumables yet because they all have an expiration date, and if we order too soon, they might be already expired. Then we also need to seek Nagoya approval. In DRC, it's approved. In Cameroon, it's being prepared. In Ghana as well, and in Burundi as well, in the wait of ethical approval. Training on sampling and the protocols and procedures was foreseen in DRC already in March, but due to unsafe circumstances in the country, we had to cancel this training, and this will be retaken as soon as possible as the situation is more safe again there. And if not, we will try to train online. We already made videos on how to sample the biopsy, and we will continue to make videos to fill up that training if it cannot be retaken. And I would like to acknowledge all the partners in the study: Marie-Josée Kaberi from INRB, Valérie Donfac from the Centre Pasteur du Cameroun, Professor Dorothy Yeboah-Manu, and Dr. Adwa Asante-Poku from the Noguchi Memorial Institute of Medical Research, Michelle Sabadoko, the legal representative of Leprosy Foundation in Burundi, and Shelta Vanzi of the Colorado State University, and also Sandeep for providing the biomemes, and also the Leprosy Research Initiative and the Turing Foundation for the funding. Thank you.
Thank you, Sofie, for your interesting presentation. Sofie, could you explain us what was the inclusion criteria for your patients?
The inclusion criteria are not having a facial lesion, because a 4mm biopsy doesn't need sutures, but it's still in the face, so we don't want the scar to be in the face. Informed consent is a very important one, and the age is also important.
Sofie, I have also a question related to Ghana. You said that they will do the sequencing later. What is the problem? Why did they not start not include it in the sequencing program?
So they have a really good sequencing center, but they are not trained yet on leprosy sequencing, and we don't have the funding yet to do the training. But we're now looking to see if they can come and do a short course at the ITM, and then we can maybe extend the stay so that sequencing can be taught here, and then subsequently can be implemented in Ghana. So as soon as possible.
Okay, thank you.
Because it's quite particular. It's not like TB that you culture and you just put it on a sequencer. The target capture is a bit more complicated.
Okay, thank you. Thank you very much for your very clear presentation, Sofie, because it's an update only because the project only started in July, but it was very clear and interesting. So the next presentation will be given by Dr. Becky Rivoire on the famous leprosy vaccine, LepVax. You have the floor.
Thank you. Hello, as mentioned, I'm Becky Rivoire from Hope Rises International, previously American Leprosy Missions. As the LepVax program coordinator, I'm excited to share our work with you today. So why develop a leprosy vaccine? Leprosy transmission continues to be a significant challenge, with approximately 183,000 new cases detected in 2023, of which nearly 10,000 had grade 2 disabilities, and more than 10,000 were new cases in children. So to truly eliminate leprosy, we need a better vaccine that provides long-lasting protection. And that's why the WHO 2030 Global Leprosy Strategy prioritizes a vaccine development as a key step towards a leprosy-free future. LepVax consists of 2 active ingredients. The LepF1 antigen is a quadrivalent fusion protein engineered with 4 M-leprae T-cell antigens. The adjuvant is a synthetic glucopyranosyl lipid A in a stable emulsion, and it is a TLR4 agonist that generates antigen-specific Th1 responses. Preclinical data have shown promise that LepVax can work both as a preventative and a treatment, offering hope for stopping transmission and improving outcomes for those already affected. Studies in mice suggest strong preventative potential for LepVax. 1 month after vaccination with either LepVax or adjuvant alone, mice were infected with M-leprae. And 6 months later, analysis of their foot pads showed a highly significant reduction in bacterial burden, highlighting LepVax's promise as a prophylactic vaccine. LepVax also shows therapeutic potential, as demonstrated in the 9-banded armadillo, one of 2 natural hosts of Mycobacterium leprae. These animals closely mimic the structural, physiological, and functional aspects of human leprosy, making them a valuable model for studying leprosy vaccine effects. LepVax immunization delayed the onset of motor nerve conduction abnormality shown in the top graph by the red line, and also reduced the number of animals with sustained neurological damage after M-leprae infection. Likewise, LepVax vaccinated animals had less cutaneous nerve damage, shown in the bottom graph, compared to control animals evaluated by CMAP, and no intraneural inflammation was found. These results highlight the potential of LepVax to protect against leprosy-related nerve damage and could support early recovery of sensory nerve function. The LepVax clinical development plan outlines a clear pathway for vaccine development. The phase 1A clinical trial was completed in 2019. LepVax was found to be safe and immunogenic in healthy individuals living in a non-endemic area for leprosy. The next step is to run a phase 1B trial in a leprosy endemic region to support preventative and therapeutic studies. Our committed development team includes Access for Advanced Health Initiative, Hope Rises, Oswaldo Cruz Leprosy Laboratory, their clinical center, and Biomanguinus, and the Sasakawa Health Foundation. This image shows our project principal investigator, Veronica Schmidt, and her team, and as well as Aya Tobiki from the Sasakawa Health Foundation. To aid in the development of LepVax, we're proud to have an international vaccine advisory group made up of esteemed experts with diverse experience. Their guidance is invaluable as we move forward. After 5 years of hard work, we're excited to share that in October 2024, the Brazilian regulatory agency, Anvisa, gave the green light for LepVax phase 1B trial to begin. Reaching this significant milestone is a testament to the outstanding effort and collaboration of the team. Unfortunately, a setback has occurred, requiring additional stability testing before the trial can proceed. The phase 1B trial aims are to assess the safety, immunogenicity, and tolerability of LepVax in high antigen doses. The trial will take place at the Leprosy Clinic at Fiocruz in Rio de Janeiro. The study will be a double-blind, randomized, placebo-controlled trial with antigen dose escalation. Healthy men and non-pregnant women without known exposure to leprosy will be enrolled. 54 participants will be divided into 3 groups of 18 for testing a low dose, a high dose, and a placebo group. LepVax will be administered intramuscularly 3 times, 28 days apart. Blood samples will be collected on day 0, 7 days post-vaccination, and 3 months after the final immunization. Immunological analysis will include PGL1 serology, quantiferon, IgG serology, and the PBMC cytokine production. To establish a baseline for evaluating candidate biomarkers of disease, in later studies, the samples will be evaluated by flow cytometry, multiplex, and gene expression. Participants will make 10 visits and receive 3 phone calls over a total of 421 days. Led by Aya Tobiki from Sasakawa, the engagement of people affected by leprosy in the LepVax trials is a unique part of this vaccine research. Morhan will contribute valuable insights on ethical standards, practical efficiency, and sharing of research findings with the affected communities. This partnership aligns with WHO guidelines to strengthen the participation of those affected by leprosy in leprosy-related services. The development of the LepVax phase 1b trial has faced several challenges, but we are steadily moving closer to launching this crucial trial. Regulatory review delays persist, but the drug dossier has been approved, and future submissions will be processed faster. Secondly, the partnership transparency has caused some delays, but changes are being made to clarify roles and responsibilities. Third, the only remaining hurdle for the phase 1b trial is the vaccine shelf life. An additional stability study will soon be underway, and the material is expected to meet specifications. After in visa review, we anticipate starting the trial by September. This trial is a key step toward assessing a leprosy vaccine that could help eliminate transmission and offer new hope for those affected by leprosy-related disabilities. We are deeply grateful to the many organizations that have contributed to the success of LepVax so far, and I acknowledge a heartfelt thank you to our team at Oswaldo Cruz, AHI, Sasakawa, Hope Rises, and the Vaccine Advisory Group for their unwavering dedication and expertise. Thank you.
Thank you, Becky, for sharing your valuable study. We have a couple of comments, not questions, on the box from Bernard Nafs, and he commented, is there any indication that this vaccine is better than the heaters, since only 20% of people are depending on the adaptive immunity against leprae? And he also commented, protecting against the infection, I mean. What do you think?
Yes, we do believe that the vaccine will provide some protective immunity based on the animal studies. I don't understand the ithers. Can you explain what that means? The ithers, since only 20% dependent.
I think he means others.
I'm so sorry. Yes, of course. Yes, the good news is that we do see that the vaccine has some protective effect in the animal studies, and that still needs to be shown in humans. So if there is a subclinical or low-level infection, we are hoping the vaccine will prevent development of disease. And for those that might not be able to develop a Th1 response, we do see B-cell response as well. We can actually track the immunoglobulin response to the LepF1 antigen.
Thank you, Dr. Becky. Any questions?
I have maybe one more question. Could you give some information about the placebo that you will use?
Yes, the placebo group will be a saline injection as opposed to the adjuvant. And the reason is because the adjuvant alone does not show any reactivity in animals, and we felt like that was not a very necessary application.
Thank you very much. Excellent. I hope you will succeed in phase 1B very soon. Thank you very much for your excellent presentation. We go to the third presentation, the WASH presentation from Dr. Sruthi Konduru.
I would like to present about holistic investigation for environmental presence of Mycobacterium leprae and its implication in leprosy transmission through WASH and One Health approach. I am the research associate in this project. Our grant support is being provided by Leprosy Research Initiative and Turing Foundation. This is my key research team, and my participating institutions are based in India: Lepra Society, Malla Reddy University, ICMR Institute of Tribal Health, ICMR Institute of Biomedical Research, the Leprosy Mission, and Hope Rises International, formerly American Leprosy Congress. India still records several thousands of new leprosy cases each year, indicating ongoing transmission. There is an environmental presence of Mycobacterium leprae that plays an important role in leprosy transmission. Inadequate sanitation adversely affects both community health and social well-being. This current study investigates the link between leprosy, environmental presence of Mycobacterium leprae, that is concerned with WASH practices and handling of livestock in India. The primary objectives of the study are to investigate and compare the association of environmental presence of Mycobacterium leprae in animals, water, and wastewater between leprosy cases and healthy controls, with reference to reported WASH behavior. The methodology of the study is a mixed method case control longitudinal study. The study period is Jan to December 2024. The study tools that have been used are COBO tools, WASH questionnaire on WHO, UNICEF Joint Monitoring Program for Water Supply and Sanitation, and focus group discussions. The study samples are like 31 each in test and control households for questionnaire, that is quantitative, and 5 in each test and control households for focus group discussion. Our field sites as shown in the map are Odisha and Andhra Pradesh in India, and the lab site is Hyderabad, Telangana. This is to the right side, this is the flow chart of the study. The case and control identification, the household contacts screening. Then it is divided into active case detection and environmental screening, and the procedure follows. The snapshot of the key project activities. There is community engagement, which involves health education, focus group discussion, and assessment of WASH practices. There is active case detection through different household surveys and contact screening for household of new and treated leprosy patients. And there is environmental screening, collection of water, soil, and animal livestock samples from case and control households. These are some of the project activities that is being initiated, orientation meeting, and there is the piloting of the tool that has happened, and focus group discussions, data collection and household surveys. The project review meeting with the investigators of the project happens bi-weekly, twice a month. The key findings of the study are: population that is covered is 49,153. The number of households that is being screened is 62, in which 31 are test households and 31 are control households. New leprosy cases that have been detected out of this are 5. The demographic characteristics of the participants are: most of the participants who have leprosy belong to the age group of 20 to 50, and most of the population are males. The key findings of focus group discussion and WASH are: focus group discussion among AIDS cases, they have lack of awareness about WASH practices, and among controls, they have awareness better than the cases. 20% of the households with leprosy use borewell water for drinking. 28.6% of the leprosy households used public tap for drinking. The odds ratio between both of them is 1.52. The preliminary observation from this study highlights a potential correlation between WASH factors and incidence of leprosy. These results indicate that the individuals consuming water from public taps are at a high risk of developing leprosy compared to those using borewell water. These findings emphasize the necessity for appropriate public health interventions. The impact and the lessons learned are active case detection, where 49,000 population was screened, and among them, the screened population, there were 5 cases who had symptoms of leprosy, and they were referred to the PHC, and the MDT treatment for them has been started. There is then the capacity of building of CRPs. They were trained on locating the test and control households using Google Maps, which they initially found very challenging to understand. A separate training session was conducted by the team to improve the skills in accessing the digital tools. The way forward for the project is continuing engagement with the community and local health departments for household contact screening and active case detection. Finish the household data collection, then environmental and animal sampling for Mycobacterium leprae, and dissemination of the project findings. These are some references from which I have taken. Thank you.
Thank you very much for these preliminary results. The questions now?
Yes, there is one question from Jessica Farley. Do you have preliminary data for sanitation findings? A study I've worked on in Ethiopia show a stronger association of leprosy and poor sanitation than leprosy and poor water access source.
We have the raw data. So you want me to share the raw data? I did not understand.
Do you have preliminary data for sanitation findings?
Yeah, we do have the data. If you could give the mail ID, I could share the data.
I think the question also asks if there is a strong association between leprosy and poor sanitation.
As of now, we have not. Yeah, as of now, we have not done the association. This is a preliminary observation. So on the further note of when the complete data collection is done, I would be able to comment on it better.
Yes, Jessica also asked, did you find association? And you have replied. Jivan Saka is asking, how much of bacterial contamination do you expect per gram of soil or per millimeter of water in the environment? It is, if it is low, will you be able to determine viability with qPCR?
I'm not that much sure about the lab results. I will get back to you on this. We have not done any, we have not done any sampling, and we have not started the initial part of it. So I can comment on it only when we have started collecting and the lab results are ready.
But could you expect to performance qPCR on those samples or just?
We have like DNA extraction and RNA extraction and qPCR will be performed on them, but about lab, I cannot comment at this point right now.
Thank you.
Any other questions?
There is no more question.
Well,
yet.
I have one question related to the environmental and animal samplage. Which kind of animals do you plan to investigate?
Only dairy animals like which are like cattle, like that. Cattle animals we are planning to investigate.
Okay, and environmental specimens? Which kind of environmental specimens?
Soil and water.
Soil and water. Okay, thank you very much, because it was only a preliminary study that started
Yes.
in May 2024.
Professor Francois, we have enough time to another
yeah,
question.
no problem, yes.
Thank you. Itu is asking, what is the method for diagnosis of leprosy?
What is the method for diagnosis of leprosy? We will do like active case detection and find the correct this for leprosy. I'm not sure. I didn't get the question actually. Could you be more specific on like what is the diagnosis means, like what you are actually asking? Can you be more specific?
What is the method for diagnosis of leprosy regarding clinical basis?
On a clinical basis, we are doing active case detection and finding like what are the clinical symptoms if there is present for like on the patients that are there, then we confirm them on a clinical basis. Like whenever we go for active case detection, so if they're having clinical symptoms suggestive of leprosy, then we confirm the diagnosis, because clinical diagnosis is currently the gold standard for treatment, like for the further treatment. Did I answer your question?
No more question. Maybe we can move on to the next presentation.
No more question? Sure.
Sure, not
Okay.
yet.
Now, Dr. Nabura Ioteba, you have the floor for your presentation.
Thank you, Professor Boteos. And greetings to you all. My name is Nabura. Next slide, please. And I am delivering this update presentation on behalf of a multi-team comprising of the important on-the-ground local Kiribati people and visiting support team members from Australia and Aotearoa, New Zealand. I want to acknowledge a couple of people, very important team members from this group. Firstly, Dr. Jeremy Hill is the Australian Pearl Team lead based and living with the locals on South Tarawa in Kiribati. And then there's Dr. Arturo Cunanan from the Philippines, who is a Pacific Leprosy Foundation consultant and WHO expert on leprosy, involved in the review of all referrals and also does the validations of all new cases of leprosy. Next slide, please. Kiribati is a nation of 3 island groups spread over the vast Pacific Ocean. You will have already appreciated its remoteness and relative isolation. The capital island of Tarawa, where the combined project is located, belongs to the Gilbert Islands group. Over half the population live on Tarawa. For almost a decade, WHO has produced data which showed Kiribati has reported the highest rate of new leprosy cases. Next slide, please. In 2018, the Kiribati Ministry of Health and Medical Services and Pacific Leprosy Foundation embarked on the STR chemoprophylaxis to household contact. This effort to control leprosy showed good acceptability and success in both retrospective 2010 to 2017 and prospective cohorts from 2018 to 2022. Although commendable, the modeling study on the right showed that in combination, a targeted household STR PEP followed by 3 rounds of mass chemoprophylaxis will reduce the number of new cases quicker and sustain the lowest level of leprosy new case findings for a much longer period. Conceptualized and endorsed by the Kiribati Ministry of Health and Medical Services, the combined project became the right vehicle for this strategy. Next slide, please. In the combined project, we set out to determine if community-wide screening, treatment, and mass chemoprophylaxis for tuberculosis and leprosy is feasible and effective. Next slide, please. The combined team activity include screening for symptoms, sputum expert PCR, chest X-ray, and tuberculous skin test before some people are referred to the National TB Program and the National Leprosy Program for further assessments and treatments. Next slide, please. Combined project did a pilot program in Nanikai, one of the villages on the main island of South Tarawa, and now it's moved to Pesio, the islets at the end of South Tarawa, following that pilot. After a slow start, half of Pesio has been covered now by the end of 2024. Next slide, please. At the beginning of 2024, you can see the green bars represent the catch-up focus activities on rifampicin chemoprophylaxis following a disruption to the supply in 2023. Registration and commencement of TPT chemoprophylaxis activities have increased substantially in the latter part of 2024 with the restoration of rifampicin supply and also an increase in staff numbers with some procedural adjustments. And on the table in the right, in quarters 3 and 4, registrations were more than 1,000 and rifampicin consents were more than 90%. Next slide, please. 9 people with confirmed leprosy, 4 new cases and 5 defaulters were started or restarted on treatment. This is a detection rate of 243 per 100,000 people, almost doubling the background rate. 2 cases were biopsy positive and all 9 cases were validated by Dr. Kunanan. Next slide, please. In terms of outputs, the program has set up a high-quality screening for tuberculosis and leprosy and contributed to capacity improvement with the use of technology. The team have engaged well with the community and showed willingness to adjust to the needs of the community. Rifampicin isoniazid chemoprophylaxis is enhancing population coverage in preventing leprosy in addition to the SDR-PEP and also the actual treatment for TB. The project has also contributed to the local and international body of knowledge. Next slide, please. The combined project has integrated basic training into Pearl staff training to detect the features of leprosy and the leprosy training module for all of National TB program training has also been developed and delivered by Pacific Leprosy Foundation, the National Leprosy Program, and Dr. Kunanan. Streamlined referral pathway for leprosy has been developed and data collection continued to be robust. Engagement with stakeholder groups, people affected by leprosy, and the PEL study team are conducted through quarterly meetings. And about 50% of the Peso population have been covered to date. This is the population of about 20,000 people. Information. Next slide, please. Looking ahead, funding from the Australian Department of Foreign Affairs and Trade has enabled the PEL study to continue through to 2027. The funding has allowed the team to employ more local nurses and community health outreach workers, which has almost doubled their numbers. There is now real potential for scaling up of activities with improved coverage and reduction in dropouts. The steering committee will continue to provide strategic advice and direction while the involvement of the LASCA group is positively anticipated. LASCA is the first community advocacy and support group of people affected by a condition, disease, in this case leprosy, in Kiribati. We're not aware of any other similar groups from the other Pacific Islands countries. Next slide, please. In conclusion, screening for TB and leprosy in a high burden setting is feasible and worthwhile as the diagnostic yields are very good. This is underpinned by developing and sustaining partnerships, harnessing knowledge and building capacity of the locals, ongoing expert input, the use of technology by the team, and also their ability to adapt. Integration of health services in settings where resources, particularly funding and capacity, are often limited, are therefore prioritized. Next slide, please. We want to acknowledge how grateful we are for the funding from Leprosy Research Initiative and the Turing Foundation, which is making this combined project possible. Thank you for listening and your time.
Thank you for your presentation, Dr. Nabura.
This is a very ambitious project.
We have one question from Chubabien. Do you have any idea of the sensible different percentage of acceptance in Q2? Was the investigation done in different communities or with different tools?
So for all the quarters in 2024, the project combined activities were all on Pesio, which is that little islet at the end of the main island. Quarter one was a wee bit slow, as I mentioned before, because of supply chain in the drugs that the team needed for the TPT treatment, and also there were a few staffing issues in terms of the questions or the tools. It was basically the same kind of tools. The only differences were later on in the quarters. In quarter 3 and 4, the team switched from a community-based kind of one setting to visiting the households and doing the screening activities there.
Thank you, Dr. Nabura. Dr. Ri Yotsu has a comment that she have been in Kiribati in 2017, and during that time, this project was only a plan. She's happy to see how it progressed. It's a strong program, good practice of integration with leprosy and TB, and she has also a question. The remote islands are a real challenge in Kiribati. How are you reaching to them?
So the combined project is going to be done only on South Tarawa for now, but the hope is by developing the capacity of the local teams that they will be able to continue on to the outer islands, which are obviously a bit harder to reach, as you know, and those will pose a fair bit of challenges, I guess. But for now, the project is going to be just based on the main island, where, as I mentioned before, just over half the population live.
Thank you. We have another question from Dr. Vivek Vasudev. What was the dose of the rifampicin and TINH administered?
As far as I know, the team or we are using the WHO guidelines.
Thank you. No more question.
No more questions?
Oh, one question more from John Figueroa. How are disability identified and managed?
Yes, that's a very, very good question. And that's always been part of the work that we do in the background through Pacific Leprosy Foundation and the National Leprosy Program. With the combined project, we only had one MB child case, but none of the 9 cases had disabilities. Or in terms of the actual kind of disability identification, there's the usual kind of pathways from the clinical management, and as we said, we do travel often with Dr. Arturo Cunanan, our leprosy specialist, and so we do kind of come across a few people with disabilities, and there's a rehabilitation center now just next, about two buildings away from where the National Leprosy Program is based, and we do refer our people there. for further management. Some of the ulcers obviously are taken care of by our clinic nurses or some of the other department nurses, the outpatient clinics.
Thank you, Dr. Nabura. There are no more questions.
So I would like to thank all the presenters for their excellent presentations. And we still have more than 10 minutes for additional questions. If you want to ask questions to some of the presenters, we have plenty of time.
E2, Dr. E2 delayed one question, but I think it's interesting. This question was for Dr. Becky Rivoire. As Leprae was a slow-growing bacteria, how do you differentiate between self-healing and respond to LepVax? I think it was interesting question.
Thank you. I tried to answer his question and I couldn't find it, so thank you. Yes, the possible trial for therapeutic application will be performed with MDT. So the new cases that are identified will receive the vaccine with their MDT treatment. And so we probably won't be able to actually know the self-healing compared to those that are healed, hopefully, from the vaccine application. However, the placebo group might give us a little bit of insight into that question. From the preventative studies that we're planning to do, the individuals will be probably household contacts or those within a certain vicinity. And so there is a possibility that there might be some that have been exposed to Leprae and starting to develop disease, but not showing clinical signs yet. And so we would have to, I don't think we would actually be able to know which ones would be self-healing versus those that would be healed from the vaccine, except for the placebo group.
Thank you. Thank you for the questions and thank you to the presenters. Professor Francois, I think there are no more questions.
I think people are getting tired now.
I think there is one question that just popped up in the Q&A box. If Lucrecia, you want to?
Yes. Dr. Givan Saggia is asking, is the LepVax activating both CMI and HMI?
We are not aware of the HMI, but we do know that LepVax activates the cell-mediated immunity when given to animals, both mice and armadillos. But what we also know in our phase 1A study that we have an IgG response from the human living in a non-endemic area. So we would expect activation of the CMI at the very least in the phase 1B trial and beyond.
Thank you. Any questions for the presenters? Say thank you, Dr. Becky. I really hope this vaccine works. Best wishes. And Malkin Sar, one additional question here. In a low endemic setting, how would you suggest to use PEP? For example, only for contacts, or would it be interesting to use laboratory tests and give PEP, for example, only for contact with positive F? I think this question is to Dr. Nabura.
Obviously, I couldn't say what would be the best way in a low endemic setting. But PEP has been shown to work for household contact in terms of using laboratory tests. I guess that's going to be dependent on the country's capacity, whether they can do field tests with PCRs or other kind of methodology that they need to confirm diagnosis for leprosy. We are very fortunate that we have our expert with us, so Dr. Kunanan, and so both clinical and we are also using laboratory means to diagnose or to confirm some of the much more difficult or complex cases. But our laboratories tests are done in New Zealand from the country. So we sent biopsies, skin biopsies overseas.
Thank you, Dr. Nabura. And another question to Dr. Becky. Which are the countries where LepVax study is in progress?
Thank you for that question. We hope to, well, initially it's in Brazil, and we will need to expand to other countries as the testing for the preventative begins, because of the number of individuals that we need and also the interest in the geographical responses. So right now we are not looking at that level of commitment yet, but certainly we'll be looking at other countries that are endemic for leprosy that we can collaborate and partner with individuals to test this vaccine.
Thank you. Another question regarding the phase 1B LepVax safety trial. How long will you follow the participants?
Participants will be followed up till 421 days after the study start, and that's a requirement for any vaccine that is tested. And so the last test blood sample will be taken on day 168, but phone follow-up will happen up until the end of day 421.
No more questions, Professor Franco.
No question? No more questions?
No more questions.
Well, thank you so much to all the presenters, and I would like also to thank my co-chair, who is a very active lady for the ANESVAT Foundation, very important for the LRI. Thank you so much, and we will close the session now.